Today I'm going to give you, we're going to talk about two topics that are somewhat related to each other, but they're not identical. And that is open science and also the fair data principles. And I think you were given papers to read, one on the topic of open science, specifically data sharing, because this is the summer of data, and that's what we're going to be mostly talking about, is how one shares and publishes data sets and why one should share and publish data sets. And then also the fair data principles. And I gave you the original paper that was published back in 2016, which has had a huge impact on communities around the world who are trying to make their data available. So before I get started, I just want you to know that I'm a professor Emerita at UCSD in the Department of Neurosciences, but we also have a tech startup that we started out of UCSD, and it develops tools and services around research resource identifiers. So for full transparency's sake, because this is an open science, I always make that known before I give these talks. So as I mentioned, our topics today are going to be open science and data sharing, data as a research output, in addition to the types of papers that you're used to publishing, the fair data principles, a little bit about why fair data was introduced, and then a deeper dive into some of the fair data principles. We'll talk a little bit about data repositories, and then the exercise is going to be for you to do a fair analysis on some of the data repositories that have been established by NIH to house data coming from experimental studies. So I'm hoping to finish up the main part soon enough that you have a fair amount of time in order to be able to do that analysis. So first, let's talk a little bit about open science and what is open science. Over on the right here, you see the little open access icon. You may have seen that on publications. Basically, open science is the movement to make science research, including publications like open access publications, but also data, physical samples, software, basically all of the research outputs that you would produce during the course of a scientific study open. And that means that it is available to anyone who wants to be able to access it. There is a mechanism to make sure that it is accessible so that people can actually acquire this. It emphasizes transparency, accessibility, right? Also, team science is a part of this. So I was just on a call earlier today where they defined open science as equivalent to team science. Of course, you can work in teams and not have anything open, but the idea that it is designed to be able to share things with other researchers and the public and others is integral to the idea of open science. So it encompasses practices such as publishing open research, campaigning for open access, and encouraging science, even to practice open notebook science. So there are some people who feel that our lab notebooks should be online and that anybody should be able to view them. And there are people who actually take that step. But I generally stop at making it easier to publish and communicate scientific knowledge. So that is the definition of open science that we're going to use. And feel free during any of this to interrupt with a question in the chat. I won't necessarily see it, but Koei can monitor it, or just unmute yourself and ask a question. So what are the major motivations for open science? Well, it really is a consequence of the digital revolution. When I started in science way back when, most of the data was on spreadsheets that were physical paper files, and you did use your own local computer. But the idea that there was a mechanism to widely disseminate any of these products, it just wasn't there, right? We used mainframe computers if we used them at all, and that meant that everything was isolated. And so, in fact, we've always had a means of knowledge distribution. It was called the library. We would publish these papers, we would print out multiple copies, and we would disseminate them to libraries around the world. And if you had access to that library, you could, in fact, go in and take that knowledge and share it with yourselves and share it with others. But with the digital revolution, it meant two things. It meant, first of all, we could distribute well beyond the boundaries of physical libraries, but it also allowed us to set up shared computation in other ways. We could expose our data to computation. So when we put things online, if we put them on in a way that allows a computer to access it, it means not just myself can go and look at it or the librarian, but I can deploy search algorithms, I can deploy search analysis algorithms across data at a much larger scale. So I believe that open science really is going hand in hand with the digital revolution and the fact that things are now accessible both to humans and machines in ways that they never were before. The two main arguments in biomedicine for why we want open science is two. One is research is largely funded by the public, and the public should have access to the results. This is a large motivation. We'll talk about some examples from the news shortly. But I have been in cases where I've had a loved one who has been ill, and I've been reading articles, and I try to send them to people, and people say, how come they're charging me $39 to access this? And NIH has taken a large step in this by mandating that any research that is funded by the National Institutes of Health must be made open access at least within a year. So the idea that the public has a right to access the fruits of taxpayers' money is a large motivation. But I think an additional argument, and one that's extremely compelling, is that we contend, those of us who support open science, that open science is better science. And I think the idea that there is transparency, science makes errors, things are controversial, but the more eyes that you have on it, the more likely it is that these errors are going to be caught. Also helps prevent fraud and other sorts of things, which are rare, but they do happen. And a large motivation, at least for me, is that, again, this increasing computational capacity that we now have can mine and put together biomedical literature and data in new ways that individual humans simply cannot. However, if our computer algorithms, and this is true in dkNET, can't get access to all of the literature, for example, or can't get access to all of the data, then we're getting incomplete pictures. So one of the big motivations, for example, of the University of California severing its ties with Elsevier recently was that if research is not open access, then we can't mine the full contents of the biomedical literature, therefore we cannot put it together. So there's a lot of motivations for why it is that we want to open it up, and they are humanitarian, but I think also they are allowing science to do its job better. And I think I have two things from the headlines, right? Using the coronavirus, I had some examples in here from the spinal cord injury that I reviewed in that paper of data sharing in psychology, but I decided to replace it with the fact that we're in a global health crisis right now. And in a global health crisis, the major publishers will all of a sudden open up their journals to be open access. And so that's wonderful and that's great, but we might ask, well, if they have to do this in a global health emergency, why are we not doing this all the time? And at any given point, people may have a health emergency. It may not be global, but it's very important to them. So you can see that they do that there's a push during times when we need rapid information flow for open access publishing. This is where, of course, again, you publish it and everybody's allowed to read it without a personal or a library subscription. So that's one of the things that COVID brought. You may also have seen the big controversy over papers that were retracted from two of the major medical journals, The Lancet, also the New England Journal of Medicine. And these were very interesting. This was done in early June, where a huge study with the headline that hydroxychloroquine is harmful and that it is harmful in a long-range study was published in The Lancet. Within a few days, however, people started to question the types of data that were available. They had not made their data available, but people questioned the finding of the studies and how they gathered their data. Within a few days after that, the journal actually requested that the data be subjected to independent review. And the authors actually of the paper requested this because they had not done the data analysis that came from an independent company. And when the researchers asked for that data, it was not provided to them. They would not give them the data. So because of this and because of the severe questions, they ended up retracting it. However, during this time, they halted clinical trials. There were several large clinical trials that were going on around the world, and they halted them out of fears that hydroxychloroquine was causing adverse effects. So there was a real impact on this almost immediately. And one might argue if the data had been made available right away, that either the peer reviewers might have caught this, but at least it would have been transparent. One would have been able to look at this data and before one halted a study, make your own conclusions about whether this is correct. So you can see that open science, even though it was sort of it has its adherence to it, I think that the COVID emergencies have magnified some of the problems with our current system. So trends in open data. As I said, I was really going to talk mostly here about open data and not open access of publications and code, but the issues are all related to each other. The idea is that when you publish a paper, you should make the data that underlies the claims in a paper available. And you see in the previous case that was not done, and it ended up to a major embarrassment. And again, beyond just embarrassment for scientific, the participants and the journal, it had real world health consequences. So the idea is that you want to publish data as a primary product of research, just like you would a paper. And you do that for transparency, you do that so people can reuse it, you do it so you can push this, pull this data together to increase analytic power, because you can do things with large amounts of data that you can't do with small data sets. Now, there has been trends in the last few years in so-called big science, where funders like the National Institutes of Health or private funding agencies or the National Science Foundation pay researchers to generate large pools of data that is freely available and more usable. So they'll often be, large studies of imaging or what have you, where they follow people over many years, and the entire study is designed from the get-go to make data available, and researchers who come into it know that they are going to have to make their research available. But they're increasingly requiring data sharing, not just of that big organized data, but again, these little individual data sets that you might produce as part of your study. And that's generally given the name long tail data, because if you do a curve like this, you see this long tail that goes out, and it basically says these are little tiny data sets, but when you add up these little tiny data sets, they actually make up a significant portion of the data that we produce. So I'm going to focus not so much on the big science, although the FAIR principles certainly hold there as well, but long data, right? What do you do for your own individual data to publish it as an actual research output? And this is actually a quote that I use all the time from Neil McKenna, who spoke to you yesterday, that I like so much. Small data done right are big data. So you actually saw some of the power of bringing together all of these small data sets in the Signaling Pathways Project, where you could do analyses across things that you couldn't do if you only had one single data set, right? So the idea is, how do we share data in a form that allows us to use the data beyond the lifetime of the study? And how do we share data in a form that allows us to more easily combine data across studies? So this is some high-level points from the new strategic data plan that NIH issued last year. So they issued their NIH strategic plan for data science. You are all young investigators, you're starting your careers. This will impact you as you move on. So NIH is getting more serious about data and what is done with it. That traditionally has not been the case. There are large federal agencies like NASA or the Oceanographic Agency, where, as somebody said, data is built into their DNA, right? They acquire these huge data sets, like from the Hubble telescope and things, and they make them all available for people because that's what they do. But traditionally, NIH had not done that, right? This was a research institute. They funded researchers. What the researchers did with their data was their business. And there's a huge shift that's going on inside of NIH now to recognize that biomedicine is very much a data-driven science. It needs to be a data-driven science. And so there's new effort, right, to treat data products more seriously than they have been done in the past and more formally. So they've outlined several things that need to be done. We obviously need better infrastructure inside of the lab. They want to have a modernized data ecosystem, and we're talking a lot about data repositories. Jeff will talk about them tomorrow. These are funded either through an institution or funded through some sort of entity, who are charged with taking data from your studies and making it available to the public, right? So they really function like the publishers for the journal, for your journal articles. You do the work, then you hand it over to another entity, and they're the ones who make this available. And we're going to talk a lot about these data repositories, or data archives as they're often called. They want to be able to support the storage and sharing of individual data sets. That is specifically called out in their mandate, and they want to be able to better integrate data across. So as you can see over the right, they say we're going to develop FAIR policies, because as we'll see, FAIR is a way that we do this. They also recognize that you need more help for data management, analytics, and tools, and Jeff is going to talk about data management tomorrow, because as we'll see, all data sharing starts with proper data management. And they also know that workforce development is important, right? They do need to expand the national workforce and data science engage broader community. And so those of you who develop data skills, there are a lot of training opportunities for you, and there will be a lot of job opportunities for you, because again, every indication is that this is the way that biomedical science is going. I should also say that high-level policy decisions at NIH are taking data sharing more seriously. So this is a large initiative in neuroscience called the BRAIN Initiative, and they basically announced their policy that says anybody funded by this has to put their data in a repository. They have to use standards in order to do this, and you have to make it available to people on the outside. In many cases, they even tell you what rights that you have to, what license it needs to be covered under. And we'll talk a little bit about that later. So this is just for the BRAIN Initiative, but similar policies, I think, are being formulated in other institutes. So the future will involve you publishing your data just like you publish articles. Oops. So the question then becomes, how do we publish data and in a way that promotes third use? So I entitled this Publishing Papers versus Publishing Data. And it's basically very simple. The idea is, as I've mentioned already, that people want to consider the data set that you produce, on which you analyze, which you use as evidence for your conclusions that you publish in your research paper, should be seen as equivalent to the paper itself, so that it needs to be handled in the same way. There's infrastructure that needs to be developed. So it says here, this is from something called the Joint Declaration of Data Citation Principles, that since sound reproducible scholarship rests on a foundation of data, that data must also be accorded due importance in the practice of scholarship and in the enduring scholarly record. And these really the data citation principles say it should be considered legitimate citable products of research, just like your papers. So you would put it in the reference list. We would have tools that could count how many times your data has been cited so that we can track it. And essentially, again, it elevates this to what people call a first-class research object, right? So we know how to publish papers so that could be understood and reused by people we don't know. There's a very standard format to the scientific paper. I once went through an exercise with somebody where they just showed a blacked out paper where all of the sections were blacked out, and he asked us, what is this? And we're all like, the title, what is this? It's the authors, the affiliation. We could tell you every single part of the section of a paper just by where it was placed. So the title always goes here, the abstract goes here, the authors go here, the introduction is here, the affiliations and contact authors are here. It's a very standard structure. And we have standard metadata, data about data, which we're going to talk about, which goes with these papers. Every paper is going to have authors, affiliations, a contact author. You can go to any paper and say, well, who's the contact author? And you know that you will find that information somewhere because all journals require it. It basically gives it a DOI or a PubMed ID, and we're going to talk a lot about these in the future. And it has a standard structure: the abstract, introduction, methods, results, discussion, figures, tables, references, and acknowledgements, right? These are pretty much the same across all journals. Sometimes they move around, and some type of papers may merge a couple of them, but you know that you're basically going to find that. Now, this didn't happen overnight. This first scientific article was published over 350 years ago, and it looked very different than it does now. But over time, we have all merged onto the standard structure. And consequently, that means if you open up any journal, if you see it online, you can read the paper, you know where to find things. People write them so that they can be understood by others, right? Nobody calls their paper Maryann's data or Maryann study, right? I give it an informative title that helps somebody search for it so that they understand what's in it and lets people know. We don't really do any of those things for data right now. And this is my little video. I hope you can hear it. It's 4 minutes long, but I think it illustrates beautifully all the things that we're going to be talking about. So I wanted to play it for you right now. A lot of these topics will be picked up by myself and Jeff. So let's see if you can hear it. Let me know if you can't. Takes a while for it to start.
Hello. My name is Dr. Judy Benign. I'm an oncologist at NYU School of Medicine.
Hello, Dr. Judy Benign.
I read your article on B-cell function. I think that I could use the data for my work on pancreatic cancer.
I am not an oncologist.
I know, but I think I could use the data for my work on pancreatic cancer. Do you have the data?
Everything you need to know is in the article.
No. What I need is the data. Will you share your data?
I am not sure that will be possible.
But your work is in PubMed Central and was funded by NIH.
That is true.
And it was published in Science, which requires that you share your data.
I did publish in Science.
Then I am requesting your data. Can I have a copy of your data?
I am not sure where my data is.
But surely you saved your data.
I did. I saved it on a USB drive.
Where is the USB drive?
It is in a box. It is in a box at home. I just moved.
But can I use your data?
There are many boxes. So many boxes. I forgot to label the boxes.
Hello again. Thank you for sending me a copy of your data on a USB drive. I received the envelope yesterday.
You are welcome, but I will need that back when you are finished. That is my only copy.
I did have a question.
What is your question? You might find the answer in my article.
No. I received the data, but when I opened it up, it was in hexadecimal.
Yes, that is right.
I cannot read hexadecimal.
You asked for my data and I gave it to you. I have done what you asked.
But is there a way to read the hexadecimal?
You will need the program that created the hexadecimal file.
Yes, I will. What is the name of the program?
Citation.
I do not know this program.
It was a very good program. The company that made the program went bankrupt in 2007.
Do you have a copy of the program?
I do not use this program anymore because the company that made it went bankrupt. Maybe you can buy a copy on eBay.
I have good news.
You again.
I talked to my colleague. She knew a person with a copy of the software.
Then why do you need me? Everything you need to know about the data is in the article.
I opened the data and I could not understand it.
If you have the program, you will find it is clear.
Well, I noticed that you called your data fields SAM. Is that an abbreviation?
Yes, it is an abbreviation of my co-author's name. His name is Samuel Lee. We call him Sam.
I see. And what is the content of the field called SAM1?
Ah, yes. SAM1 is the level of CXCR4 expression.
And what is the content of the field called SAM2?
That is logical if you think about it.
What is the content of the field called SAM2?
I don't remember.
What about SAM3? Is there a guide to the data anywhere?
Yes, of course. It is the article that is published in Science.
The article does not tell me what the field names mean. Is there any record of what these field names mean?
Yes. My co-author knows what the content of SAM2 is, and SAM3. And SAM4.
Can I talk to your co-author?
I'm not sure.
I would very much like to talk to your co-author.
Well, he was a graduate student. He went back to China 2 years ago.
Can I have his contact information?
He is in China. His name is Sam Lee.
I think I cannot use your data.
You could check the article to see if what you need is there.
Please stop talking now.
Okay. So, oops, let me share here. So I hope that you enjoyed that little vignette. So that was our, oops, let me go back now. So let's go back to my final slides, or the second part, which is really dealing with FAIR. If I can get back. Okay, so the FAIR data principles were issued in 2016. They were actually floating around before that in an organization that was called Force 11, the Future of Research Communications and E-Scholarship. And I was very involved with that. I was the president of it for a while. And when people came to me and they said, we have these really nice principles, I say, you know what, I think this is going to be big. Let's post it on the website. And in fact, they have took off. And a lot of the people who were involved in this were, in fact, a part of this organization. And you can see by its name, it was really dedicated to researchers, biomedical, earth sciences, everybody who came together around 2011 and said, you know what, we could do things differently now. We could do things differently. Our current system was great for 350 years, but we could do much better now. And we want to be able to transform it. Of course, the how is hard, and also dealing with the fact that there's a huge inertia, right? It's very difficult to affect cultural change, as we all know. But FAIR really kind of put together in just a very easy acronym, a pleasant acronym, right? We all want to be fair, what it is that you need to do if you want to make your data available. So it basically said that if you want to make data a primary product of research, you want other people to be able to reuse it, then you need to make it findable, accessible, interoperable, and reusable, right? You need to make your data fair. And FAIR is quite clear that it's not just for humans, but it's also machines. And it's not just for machines, it's also for humans. You actually have to make it fair for both. Machines have to be able to access it, to process it, and need to understand it, in quotes, but humans also provide important oversight, and humans are the ones, of course, who are making sense of this, and so they need to be able to understand it as well. Both of them have to do it. So I want to spend a little time talking about some of the key concepts in FAIR, just in case you haven't had these before. One is the idea of machine readable. What does machine readable mean? Sometimes you'll hear machine readable versus machine actionable. I kind of use them interchangeably, even though there are subtle differences. But the basic idea is that data or metadata, and you often see this meta here just to indicate that it's both data and metadata, and we'll talk about those definitions in just a moment, is available in a format that can be easily processed by a computer without human intervention while ensuring that no semantic meaning is lost, okay? It also means that it is actionable, that a computer can compute on it. So if it's a date, and I have two dates and one is before the other, the computer knows that it's a date and it knows that this one is before the other, right? And how you structure your data and make it available very much influence whether it is machine processable or not. There's two types of machine readable data: human readable data that is marked up so that it can be read by machines, and HTML is a classic example of that, right? You're not looking at anything but bits and bytes on your computer, but there's a language that says, I want you to make this bold, and I want this to be the title, and I want this to be red, and I want this to be blue, right? And any web browser knows how to read that and interpret it and show it to you so that it is consistent across. And then there's also data formats, right, that are intended for processing by machines. So CSV, RDF, XML. This is the data elements themselves. The date example that I gave you is a good example. That date is not a free text field that a human understands, but a computer says, oh, this is a date, so therefore this is the month, this is the day, this is the year. And I know what types of operations I can perform on that. I know that 1960 is before 1970, for example. So it basically is machine processable. Again, these formats are only machine readable if the data contained in them is formally structured. So exporting a CSV file, and for those that is a comma separated value, or basically a spreadsheet, so an Excel spreadsheet. Exporting a CSV file from a badly structured spreadsheet does not make the data machine readable, and Jeff is going to go over some of that tomorrow, okay? So that's one of the important things, is machine readable. And here's just a really good example that I use actually to illustrate this, because I do see these examples when I, as an editor, and also when people submit data to us. So look at these two spreadsheets, right? This is comma separated value. So it basically means that we have subject, subject group, species, age. But again, think of it as an Excel spreadsheet or a Google spreadsheet. And we can look at the top one and we can look at the bottom one, and they look fairly identical, okay? However, let me escape from this for a second. If it will escape. Oops. There we go. If I actually look at this file, I see that the top one is actually an image, okay? So you notice I can't select anything into it. I can kind of click on it, but that's all I can do. So this is a picture of a spreadsheet. And before you laugh, again, as a journal editor and also in a lot of our data projects, people submit to us data as pictures. They take a picture of their spreadsheet because to a human, it's perfectly clear what this means, right? Humans can read both of these. This, however, on the bottom is an actual spreadsheet. So you can see that I can select the text. Age is a number, it's an integer. So we know what that means. Again, I can add them up. I can perform operations on them. I can take an average. I can sort according to male and female. I can't do anything here. This is a binary format. It's pretty much opaque unless you use really, really fancy optical character recognition and other sorts of algorithms. So in this case, again, we have something that is very much human readable. Human has no trouble looking at this whatsoever. This is true of almost 99% of the tables that you see in papers, right? Humans can look at them, but you can't really do anything with them. This, on the other hand, is a spreadsheet. It's linked to a spreadsheet actually in Google Drive. And you could go and manipulate this data. You could resort it. You could do whatever you want. Okay, so that's what we really mean about machine readable, machine processable, and there is a difference, and it is a common mistake that people make. So that is integral to FAIR, right? We want the computers to be able to access as well, even as we also want the humans to be able to understand it. The other key concept is metadata, and this was mentioned in the data sharing and psychology paper if you had a chance to read it, but it's a very important concept. And as big data came into existence, if you'd had asked people back in, I would say the early 2000s or 1990 what metadata is, if you ask them on the street, most people would go, I don't know, unless they were a classical scholar so that they knew that meta meant about data. But all of a sudden, all kinds of people started to talk about metadata, right? Once big data came in and the sort of data revolution in data scientists. So metadata, very simply, is just data about data. And some of that can be very structural. The file format is a type of metadata, right? This is a CSV file, this is a TIFF file. The file size can be metadata. But what we're talking about a lot here, this type is really easy to get. You know that they're file extensions. Most programs, when they save something, they will save it as a specific format, and they'll put that file extension on. And as long as you have that, most computers can interpret it. They can tell you the size of the file. They can tell you all of that. But the thing that we mostly talk about in science, and which was illustrated in that nice little video, is descriptive metadata, right? This is something that describes the data in a way that increases understanding. So it helps with discovery and identification. It can include simple elements such as title, abstract, author, keywords, but also deeper metadata such as the study organism, the disease or condition studied, the experimental parameters, the techniques. This is all of the important information that provides the context for those bits and bytes that the computer is able to process on, right? So it is very critical that we understand what it is that this thing is about, and that all our metadata is clearly understood, right? No SAM1, SAM2, SAM3. So now let's take a closer look at FAIR. And I always put this slide, there's an old saying, right, that it's like mom and apple pie, right? Nobody's against mom and nobody's against apple pie, and nobody's against FAIR. And people use that word all the time. They're like, of course we want to be fair, and we want our data to be fair, right? And oftentimes, nobody ever gets further than the findable, accessible, interoperable, and reusable part. So they know the acronym, they kind of have a general idea about what it means. But the FAIR paper, the FAIR data principles, actually went into a little bit more detail about what makes something findable, what makes something accessible, interoperable, and reusable. And as with most papers that were published, people since that time, because that's already 5 years ago, 4 years ago, are saying, well, but there's more. And we know that there's more. But the authors of FAIR basically said, these ones we know are important, and you should pay attention to these. And we're going to tell you a little bit about how you could do that. So you can see that findable has 4 principles or sub-principles, attributes, whatever you want to call them, like being assigned a globally unique and persistent identifier. Accessible says, well, you have to be able to retrieve the metadata by their identifier using a standard communication protocol, and that protocol must be open, free, and universally implementable. Interoperable means that you need to do things like use languages that are interoperable. So if you use some custom format that nobody can understand or a custom language, it's very difficult for anything else to interoperate with it. If it's broadly accessible, that gives you a much better shot. You should use metadata that use vocabularies that follow the FAIR principles. So this is the way we can get around the fact that I don't understand what SAM1 means, right? There are ways to make it easier for computers to understand human vocabularies, and I'm going to talk a little bit about those in a minute. And reusable deals with some very simple things. Like one of them is that you need to release your data with a clear license so that when people come across it, they know what they're allowed to reuse it for. And we're going to talk a little bit about that in just a moment. But that you also describe your data with a plurality of accurate and relevant attributes, right? So that you provide this rich context that we're sort of talking about. Okay, and it's very important also that if there are domain relevant community standards that you follow those. And I think Jeff's going to do some of that with you tomorrow. So I didn't want to go into all 15 of these. I thought that that would be a little interminable, but I do want to talk about a few of them, the ones that are the most closely identified with FAIR, and some of the things that we can do right now fairly easily. There are a lot of them that actually require a lot more work, but as the groups that have been involved in FAIR says, there are some things that you can do right now. So even though if you can't be fully FAIR, you can be somewhat FAIR. So in findable, number 1 is that metadata are assigned a globally unique and persistent identifier. And I think that most people who would look at FAIR would go, huh, why is that the first thing? The second is that data are described with rich metadata. So I'm really going to look into this in just a little bit more detail and show why they are important and also why they go hand in hand. So, right? Principle F1 is arguably, this comes from the FAIR people, the most important because it will be hard to achieve other aspects of FAIR without globally unique and persistent identifiers. It's globally unique and persistent identifiers that let us reliably find data that has been referenced someplace else. So if it's been referenced in a paper, or if it's been referenced on a website, it is this globally unique and persistent identifier that lets us go and find it. So hence compliance with F1 will already take you a long way towards publishing FAIR, because if you can't find anything, then all the rest is pretty much moot, right? You can't access, you can't reuse it, and you can't interoperate. So basically what it's saying is that data should live at a stable address. So you all know URLs, right? URLs are globally unique. So when you go to that URL, you can say that only points to that one web page and it doesn't point to any other web page. However, URLs are not really persistent, right? They change all the time. Websites move, you kind of take them down. So we said, no, right, that's not good enough. It has to be both unique, but it also has to be persistent. It has to be reliable, okay? And as we like to say, this doesn't happen magically by the machine, right? This is where the humans come in. Things are only persistent and unique because there are organizations that stand behind them that issue these identifiers and say, this is unique. And I'm going to make sure that if my web resource moves and I change the URL, that I am going to tell the people who maintain these registries that it has moved so that it can be reliably found. These identifiers can also be resolvable. And by that, we mean you can plug it into a web browser and be taken to the object. So why are these so important? Both, it provides a stable home for these so that you can reliably access it even when the underlying technology and file systems and everything change. But it also, because it's a stable address, allows a digital object to be reliably tied to its metadata, okay? And what do I mean by that? Let me show right here, right? So there are actually a lot of examples of globally unique and persistent identifiers. The ORCID that we asked you to get is one of those, right? That is the identifier for you. ORCID never gives the same researcher the same, or two researchers the same identifier. And you can change location, you can do anything you want, but your ORCID stays the same, unlike your institutional email address, for example. RRIDs, which Anita mentioned, are also globally unique and persistent identifiers. The ones that are most closely associated with articles and data sets, which you probably have come across before, is something called a digital object identifier, or a DOI. So every article that is published has one of these DOIs. You see that in that little funny text that appears at the very end. And this is maintained by an organization called Crossref that is supported by all the major publishers, right? So they're the ones who ensure that these DOIs continue to resolve, and they build services around them, digital object identifier, right? By globally unique, again, we mean that it identifies one thing only. They may not be reused. So sometimes you go to a website that let its URL or domain name lapse and it goes to a different one, right? This cannot happen. It always must resolve to that object. And as I mentioned, this is issued by these registries, Crossref, the RRID project, ORCID, who ensure, who stand behind and make sure that these things still work and work as the way they're supposed to. So the ORCID, as you can see, or globally unique, right, is a very useful thing for disambiguating human text. So here's a bunch of papers that come from me. They're all M. Martones, and some of them are ME Martones, and some of them are not M. Martones. And if you know that I kind of come in neuroscience, you might say, well, okay, this one here looks like a neuroscience paper, so that's probably you. But I don't know, you're involved in community science. This could be you, only it's not me. It's a different M. Martone. This one here says traumatic brain injury. Well, that could be me, right? Because it's neuroscience, but it's actually my cousin Maryann and not me at all, right? And this one up here is also me. So when you just use your name, even I myself, who should know better, am not consistent about it. Sometimes I use ME, sometimes I use M, sometimes it's Maryann, sometimes people spell Maryann as one word, sometimes they break it up, right? Names are hard. And you saw that in that data sharing vignette there, where he said, my name is Sam Lee and I live in China. It's like, well, okay, that doesn't help, right? That's not going to narrow it down for me. So ORCIDs provide a way to disambiguate names by giving it a unique identifier. Computers love unique identifiers. They can process them very quickly. But it's also resolvable, right? So when I click on my ORCID, if I put that in my web browser, I go ahead and I open it up and it's going to resolve to my profile. And you can think of this profile as metadata, right? It's like, well, here's her biography, here's websites, here's her employment, here's her education, here's all of her publications down here, right? Here's her funding. So it basically resolves to metadata that says, not only is there this person called Maryann Martone, but when I come here and I look at it, I said, this is all the information that has been tagged to that identifier that can then be brought into this central place. And that gives us some trust, right, that the computer is resolving to the thing that I thought it was, right? Because when I look at this, I don't have to guess whether the computer made a mistake, because mistakes do get made in databases and things, but I look at it and I'm like, oh yes, right, this is the Maryann Elizabeth Martone that I know about, right? So it allows you to bind metadata together about these objects, and when it is resolvable in the way that we talked about, it lets you actually bring you to those particular objects, right? Gives you a location. So the second thing in FAIR is that it should be rich metadata. What you just saw about Maryann Elizabeth Martone was fairly rich, right, in terms of my academic career. It brought together a lot of different things about me. We want, when we describe objects, to have enough metadata that we can kind of understand what it is that we're looking at, just like we said, and also that we're sure that we were taken to the correct thing, because it provides us context. So here are two data sets that are on the web coming from two databases. This is in the field of neuroimaging, and I just like to show them to someone, right? So here's number one, where you look at it. Just give it a few moments to resolve. And it's something, it's got a title, right? It's called Phantom DICOM data, and it's got an identifier, and it's working very slowly, but here it is. This is the landing page for this data set, right? This is where it takes us. So if you kind of look at that, does anybody understand what this data set is about? I'm assuming the answer is no, right? Because in fact, it doesn't give you very much information at all. Now I'm going to take you to another database of also MRI data. This is called Open Neuro. And now you see that there's a lot more metadata here, right? It gives you a title, it tells you who uploaded it, it gives you its file size, it tells you a little bit about the data, it tells you who the authors are, it gives you the file structure. So you can see in this case, this is much richer metadata. It would be a lot easier for me to find this data set because instead of being called phantom DICOM, it's called something that's meaningful and understandable. And it also, when I get there, it's like, oh, you have now told me a whole lot of useful context about this so that I'm much more able, right, to know that I have found it. And then also I'm much more able to be able to reuse it, okay? And there are attempts in biomedicine in particular to make this type of metadata standard, just like we have for articles, right? So when we have both of these, together, right? Oh, the other thing I wanted to talk about was structured versus unstructured metadata. So this is another term that I want to introduce. Structured metadata or data is data organized according to a data model, an abstract model that organizes elements of data and standards how they relate to one another. And that may not make a whole lot of sense, but I will show you an example in a minute. Unstructured metadata is information that doesn't have a defined data model or is not organized in a predefined manner. And the classic example of unstructured metadata is text, right? You can read text, but you know that if you want a computer to go in there and look for things, you need all kinds of special tools to be able to do that, right? Because it's free text. It has no easy structure that the computer can latch onto to make, to do computations. So again, here's just a classic, simple example. In a text of a paper, you might say subjects comprised male, N equals 6, and females, N equals 6, C57 black 6 mice, age 25 days. Perfectly human understandable, right? In a structured set, you would have a database that basically said there's a field called organism and its value is mouse. There's a field called age and its value is 25, and this particular thing is an integer, not free text. And here is the unit, here's the strain, so on and so forth. And when you have it structured like this and appropriately tagged, right, the computer can operate on it the same way that a human can operate on this without too much difficulty. Oops. So again, what can we do when we have identifiers and structured machine readable metadata? Identifiers can be used to reliably take us to a place where there is metadata about that object. And if that metadata is structured, then it's able to reuse it in a different context. So just to give a very simple example of this, you've probably all used online reference managers where you have plugins like Mendeley and other things, right, that come to a page. In this case, this is the article that I gave you to read. I come over here to PaperPile, which is an online reference manager, and you can see it automatically, because of tags that have been embedded in the code, says this is a paper. This looks like a paper, right? It recognizes that it's a paper because of that standard structure. If I say add to PaperPile, right? When I come to PaperPile, which is my reference manager, it's able to say, oh, look, here's the title, here are the authors, right? It basically knows that this is the title, this is the authors, this is the journal it's in, this is the abstract. It doesn't do this through any kind of magical AI. It does it because inside that page, they've structured the metadata and they've embedded those tags, right? So they've made it very, very easy for a computer to be able to process it. So that's just a very simple example of structured metadata, okay? Let's move on because I know we're a little late. Accessible. Metadata are retrieved by their identifier using a standard communications protocol. And I think you've seen some examples of that already, right? It basically says, if I take that DOI and I put it into a HTTP, a web browser, it knows how to resolve it, and it takes me exactly to that object. And this is different than search, right? So I can take this DOI and I can search it. Just basically plug it into the Google search bar. So the difference between putting in the search bar versus your browser. And you see that if I search that, it gives me a lot of things that reference that search string. So that paper's been cited and it's used in a couple of different places and it tells me all about it. When I say that it's resolvable, though, I've plugged it into the HTTP protocol, which is the web protocol for returning that information. You can see the DOI right here. And it doesn't take me to anything that's talked about it. It takes me to the object itself. So it takes me, in this case, to this particular paper that was published on RRIDs. So that's the main difference between using it as search versus resolution. Resolution means it goes to the address of that object and returns all the metadata to you. Okay, so I hope that's a little bit clear. I know there's a lot of information here. Interoperable. We're almost done, don't worry, and then we'll take a little break. Interoperability has several things in it, but one of the most important things is that the data and metadata use vocabularies that follow the FAIR principles. So what do we mean by this? The typical research scenario, let's take a look at what typically happens. And you saw that scenario in the little vignette that we showed, right? 3 different laboratories study the same transgenic mouse model of polycystic kidney disease. Now, it turns out that this mouse model has a research resource identifier, so it's got a unique identifier. But what happens when you don't use those identifiers inside of your database? You get something that looks like this, right? I've got 3 different data sets. I made these up, okay? Gene expression in peripheral organs, gene expression in major organs, physiological measurements of kidney. So here in data set 1, I see that they use this organism here, and they use the official name that is given by Jackson, and that's very good. These guys decided to call BPCK negative, and these guys decided to call it ABPCK del. These are all referencing the same object. Would you know that even as a human? Probably not. The computer certainly doesn't know, right? Because these are not unique identifiers. These could in fact describe a whole bunch of knockouts. They could describe a whole bunch of things. If you instead had used the identifier, right, the computer could say, you can call it whatever you want. You could call it M. Martone, M.E. Martone, Maryann Martone, split up names. I don't care. As long as she has the same identifier, I know that I can retrieve it, right? So it enforces something that makes it very easy for a computer to know that I could put these 3 data sets together because they're all in fact being used by the same object, right? Same with gene names. You would think that we would standardize on these right by now, but there's a historical context. Each one of these has multiple different names. There's many different ways that you can reference them. But if you use identifiers for gene names, it becomes much easier for the computer to be able to actually access it. So essentially, again, all of these things have their own unique and persistent identifier. Same with anatomy terms, right? We have cortex, renal cortex, cortex of the kidney. These 2 refer to the renal cortex or cortex of the kidney, but this could be the cerebral cortex. This could be the cerebellar cortex. You really don't know. It makes sense in the context of that database, but a computer could not go and say these 2 are the same thing, okay? There's another practice that I think also impacts interoperability, and FAIR is actually fairly mute about this, but we see this a lot in biomedicine. When people report on things, they say, oh, well, we studied subjects aged 15 to 30 days. And they put that in their spreadsheet. They're saying everybody is 15 to 30 days, so that's all you need to know. The problem is over here, they said, oh, these animals are 10 to 20 days. And they said, these are adult. So without knowing what adult is, how do I know that in fact it falls within one of these ranges or not? In fact, I don't. So I don't know that I can combine these data. Also, what if some major event happens on day 14? So does that mean that I can combine data from animals that are 10 to 20 days with 15 to 30 days, or can't I? So one of a really good practice for you is to always keep subject level metadata, always keep the animals and the information about them separate, because then if I knew that I couldn't combine animals that were younger than say 14 days with those that were older, I could select the ones that I want, right? So there's all kinds of things that we do that impact interoperability. So again, identifiers, right? We use things, they're often called ontologies and others, that allow us to be able to unambiguously identify the things that we are talking about within data sets. And if they are generally used by multiple people We all use the same identifiers. That means it's much easier for us to be able to combine these together. So that's really what the FAIR principle about vocabularies mean. And I just have a few more to go through and then we'll take a short break and then we'll do our exercise because I want to make sure you have at least 20 or 30 minutes for that. So reusable has several, as we've talked about. One is that you have a plurality of accurate and relevant attributes, and I think we've already kind of talked about what those mean. That they meet domain relevant community standards, and Jeff's going to talk about that tomorrow, so I won't go too much into it. But I do want to talk a little bit about licenses. This is one of the simplest things of FAIR, and one where a lot of biomedical repositories do not do it. And it was not the tradition. These things came about from different, it rose from different communities, and it has not been standard practice in biomedical repositories to tell you the license under which it is reused. Well, why is that important? There's a very nice statement that I like that says legal uncertainty interferes with the production and reuse of research data. So when you put a clear license on, you signal to people that they are allowed to use this data and under what circumstances they're allowed to use data, okay? So there's been a lot of effort to come up with licenses that people can apply to data that let people know, even if it's open, it's only open under certain circumstances. And when you use one of these standard licenses instead of a 14-page data use agreement, it also means that a computer would know this is covered under a license that I can go and just access this data and I can use it. Okay, so we don't really have time to go into all the different kinds of open licenses and things, but just recognize that just because something is out on the internet and it's so-called open, it may not be fully open in the way that proponents of open science mean it. When open science people use the word open, they basically mean that it is free for you to use, but it's also free for you to distribute. So if you want to give a copy to your friend, you can. If you want to build it into another tool, you can. And other people are free to sort of take those products. Not all licenses let you do that. There are some that basically say, you know what, you can use this if you're an academic and nonprofit, but you can't use it if you're a commercial company. CC BY is a very common license which says you may use it, but you must attribute the source. This is academics' favorite license. And in fact, there are a whole bunch of flavors, and licensing is a gigantic issue. But I bring it up because, again, as you start your careers, you should understand that there are these different type of licenses, and science has gotten into trouble in a couple of places where it thought it was making something open, but in fact, the license said, no, it's open for a human, but it's not open for a machine, and so on and so forth. So there are a lot of subtleties there. Okay. So are you feeling a little overwhelmed? I think you might. FAIR is a whole lot of stuff to worry about. And if you're sitting there going, how am I going to do all this? How am I going to do all this? I don't blame you, right? But I like this old Star Trek. It says, when you're waist deep in tribbles, it's a bit difficult to remember that your original objective was to guard the quadritriticale, right? So your original idea is to make sure that you need to publish your data in a form where other people can find it, access it, and then use it for the purposes that they want. So the good news on that is that I like to call that there's something called a FAIR partnership, right? And that means that the responsibility for FAIR is actually divided amongst multiple parties. Nobody is responsible for all of it. In the center are these data repositories and data registries that I referenced, right? These are the publishers of data. So they are the ones that need to give persistent identifiers and machine-based accesses and clear licenses and machine-readable metadata. That is not something that you yourself need to be concerned about if you are producing data. So the bulk of FAIR really focuses on these repositories, and they have to be ready to support it. But that doesn't mean that you're off the hook, right? You as researchers have the primary responsibility of managing your data inside of your laboratory in a way that is going to allow it to be shared effectively later on. Because if you don't acquire the metadata, or you don't have a data dictionary, right? Or the person leaves the lab, or it's on a thumb drive or anything else, right? Then there's very little that anybody can do to recover all of this. So FAIR data starts with you, but the responsibilities are quite clear. You need to have good data management practices, which Jeff will go over tomorrow. You need to have rich metadata, right? Which is part of this. You have to have all of that information available when the repository asks for it. Prepared to share means that when you go through your regulatory boards, like your IRB or your animal committee, mostly your human review board, and with your colleagues, that everybody understands when this data is acquired, that it has been acquired with the intent that it is going to be made as open as possible. Now, I didn't talk about human data, but there's a nice phrase that I had on a slide, which I removed: as open as possible, as closed as necessary, right? Some data has to be restricted access because everybody can't see it. It contains sensitive data, private health data, right? So I'm not really talking about that. I'm talking about you being able to put as much of this out in the public domain as is consistent, right, with good scientific practice. But that means that your colleagues need to know that, right? Oftentimes somebody gets all the way to the point of submitting it and their colleague says, I didn't agree to this, right? You need to prepare to share. You need to think about things like open formats for exactly the reason that you saw in the video, right? But the most important thing that you need to do is not to steward your own data. Do not put it on a lab website, right? Do not give it to your friend to put on their lab website. You should submit it to a repository who is charged and appropriately funded to make sure that these things last in perpetuity, okay? So that's the good news, and you're going to see a lot of that tomorrow with Jeff. So I'm going to take a 5-minute break, but dkNET has tried to be helpful in the area of FAIR. We're going to put out a lot of different materials about what is rich metadata, so on and so forth. But one of the things that we have done is we've created a list of community repositories. These are repositories that NIH has set up, right, who are going to steward your data. They want you to submit data to them. So very good. I don't want to keep you too long, but I hope that you found it a worthwhile exercise to start looking more deeply in some of these repositories. And as I mentioned, tomorrow, Jeff is going to take this from your perspective, not the perspective of the repository, and understand how you can help produce fair data, both for yourself, as somebody noted before, and for other users. So I think Kawei is showing you the reading list, and
the reading list for tomorrow from Jeff. Yes.
Yeah, and you know, also again, the course is going to continue. I know you're getting an overload of material, but if there are topics that you'd like to pick up some more and have a discussion on, open science, fair, whatever it is, please let us know, because we're happy to put in some additional sessions. I know a lot of people are often interested in open science, so I'm happy to talk about it further. Just let us know. Okay, thank you, everybody. Bye.
Bye.
Thank you. Thank you.